Hello and welcome to this week's What Were They Thinking? This week includes news on the Delta variant of the CCP virus, how blood tests can tell you just how stressed you are and give you a much better idea as to what effect it's having on your body, the development of an oral and, frankly, easily consumed cholera vaccine, an interesting case study, and why camels can go without water for as long as they do. The timestamp for these and other news items can be found in the description box below. Starting with the Delta variant of the CCP virus. It has been a relatively recent emerging variant of the CCP virus, yet in that time it appears to have infected over 96 countries. That spread in and of itself is impressive. What's more worrying is that the number of people who are dying and being infected by it is skyrocketing by comparison to the previous, and to some degree the original variant of the CCP virus. For example, in America alone, in the last 10 days or so, the Delta variant has now started to account for 30% of the total sequenced cases of the CCP virus. Roughly a third of all patients are infected with just one strain. A second strain of concern is the Gamma variant, which has come from Brazil, which is also extremely infectious and, at least in theory, more dangerous as a consequence. Two variants which were able to start gaining traction, largely because populations either couldn't be properly looked after and cared for to prevent its spread, or vaccinated to prevent infection. The worry with the CCP virus is not just that it infects people, but realistically, the more people that are infected, the more people who are likely to die, and, as we well know now, have long-lasting changes. Some of these are far more significant than others. We've spoken to some length on the heart, lung, and brain damage seen. One that we weren't aware of until this week is changes to blood. Being infected, or at least experiencing symptoms, for a long time after the resolution of the CCP virus is something reasonably common, and why wasn't entirely clear. This could possibly explain it. It all begins with the inflammatory response and a bunch of coagulation problems. Some of these are being seen at present with vaccine responses. But prior to that, the CCP virus was also causing similar issues. There were things like Kawasaki's disease in children. There were blood clotting disorders in older adults. All manner of problems in the blood vessels that would then increase the death toll from it. Those side effects aren't so surprising when you look at it. It's well known that they're all involved in the immune response and the ability to get rid of the virus. What wasn't so clear is that red blood cells in those who have been infected will vary wildly in size and that they show significant stiffness compared to normal red blood cells. This is going to be important. Red blood cells need to be able to have movement in them. They get squished through tiny blood vessels called capillaries. If they don't have some movement within them, they'll wind up getting stuck and blocking blood flow. This is very much like clotting, and we see it in heart attack or infarction, where blood flow stops and so does the supply of oxygen. Further to changes seen in red blood cells, there were changes in white blood cells as well. The white blood cells varied a lot more, and they're more interesting but nowhere near as clear-cut. For example, lymphocytes had decreased stiffness, monocytes were larger. Neutrophils had a lot of different changes and deformation. Neutrophils could also be seen to have odd and different changes, particularly for something that's very short-lived. But that also means that the effects seen are coming through over an extended period and being applied to each new generation of neutrophils that come out. Something that obviously isn't just something that affects the current neutrophils when the person's infected, since it keeps going long after they've recovered. With the rather depressing discoveries around the CCP virus out of the way, we can move on to more interesting experiments involving blood. This time, quick testing of stress hormones. As a general rule, the more stressed you are, the more hormones your body starts to pump out. For example, cortisone. This helps the body to conserve energy, salts, and similar. Things the body thinks are essential, and at least in evolutionary terms, were often much harder to access than they are now. Think about what your body's response would be if you suddenly found yourself in a position of famine. There's no food available. Salt is in short supply because your food hasn't been processed. Water isn't readily accessible. All of these things are important, and they are conserved by cortisol, among others. That is where a new microchip comes in. This can measure cortisone in particular, and this would be far cheaper and easier than most of the current lab test methods. They normally rely on some sort of combined molecule. One part will attach itself to the cortisone or cortisol, and the second part is a probe or marker that they can then measure the intensity of. In comparative testing, this method was just as good as the current gold standard of an ELISA assay. They're also relatively non-invasive, can be done very quickly, and don't necessarily require a clinic. That means it can be done at home with fast results. It's very similar in some ways to the current ease of testing glucose levels in blood for diabetics. Going now to diseases, but very different diseases to what we're familiar with today. In this case, the Black Death, Yersinia pestis, plague, whatever you want to call it. The Black Death, as it's most commonly recognized, really kicked off in the 1300s. This was, at the time, a massive thing. Whole cities were locked down. You weren't allowed to leave or enter. It destroyed sieges or made cities fall to the besieging forces. There were all manner of desperate efforts to try and stop its spread. Nothing seemed to work, and it could entirely clear out a city of every living being. It reduced Europe's population to the largest extent seen so far, more so than World War I and World War II combined. That is an amazing figure when you think about it. But overall, we know relatively little about the history of the plague. At least now we have some idea as to the earliest possible time it could have occurred. About 3500 BCE. The reason they can track this particular disease back to such a relatively specific time frame is they can look at the DNA and what genes are present and not present. The researchers note that it's important that the earlier versions of the plague genome lacked the means of being able to be transmitted by fleas. Fleas came on rats, which then came to Europe, and that is how the Black Death spread. Since they didn't mutate at least for 1,000 years between the infection of this person who died and the first cases in Europe, it appears that that's exactly what it needed. Something to use as an intermediary species to then get its way to people. Over 1,000 years, it acquired the necessary genes, and that led to the ability to benchmark when and where various strains of the Black Death appeared, and just what they had available. And that's what this research finds. There's a 5,000-year window in totality, and that it's only at certain points that certain genes are present, and that these affect how infectious it is. Exactly the same sort of phenomena we're seeing today with the CCP virus. Going from diseases that we know how to deal with but have plagued us for a long time, to diseases that we can deal with but continue to cause problems across the world, and how now exactly we can treat them, or at least could, if the next phase of human clinical trials are successful. Cholera is a disease we've mentioned in the past, notably in the early days of epidemiology in London, where it was found that feces found its way into the table water, which was then accessed by pumps and wells, and people would then consume this water, which created an infection cycle for cholera. This was otherwise known as the fecal-oral route. Cholera still, despite having known about it for so long and how to stop it, continues to spread and infect people. There are thousands and thousands of avoidable deaths every year. The majority of these are extremely young children and extremely elderly people. This is how the development of a rice-based vaccine for cholera could offer significant improvements. In many of the places where cholera is endemic are also countries and locations that don't have access to advanced medicine, or in some cases simply can't afford it. By growing the medicine as food, they're able to provide that same degree of protection without necessarily needing to bring in a huge amount of extra resources, specialization, and facilities that otherwise aren't going to be available. The other advantages to this include things like transport, logistics, and storage. If it's in a grain like rice, it can be stored with relatively little requirements, no refrigeration or freezing for instance. All you need do is plant out the rice, let it grow, and then harvest it. The way this works is fairly straightforward. Like with a vaccine, there is a small amount of something known as an antigen within the rice, in this case the cholera toxin B. By genetically modifying the rice to produce this same toxin in small quantities, the body takes it in, identifies it as a foreign pathogen, and the immune system mounts a response. It's exactly the same idea as if you had received a vaccine via an injection. The only difference is that you're consuming it as a foodstuff as well. The only downside to delivering this vaccine via the GI tract is that it is complicated, far more so than if you were to inject the vaccine, and this is something that the researchers have observed in their study. About 4 months after the end of the study itself, during follow-up research, about 30 people who had been vaccinated had little to no immune response. This means the protection had worn off. Next is another medical advance, but this time not vaccines, but related to spinal cord repair. Damage to the spinal cord is significant. In the worst case scenario, it can lead to quadriplegia or paraplegia, if not death. In other cases, it can cause chronic, long-term, and possibly lifetime debilitating pain and limitations on physical movement and sensation. If you're going to look at the spinal cord in terms of what it does, it's a bony shaft with the spinal cord inside. Inside the spinal cord, you have white and grey matter. Along the entire length of the spinal cord, nerves come out and connect to the body in various ways. This is why, depending on where the spinal cord is damaged, you'll see different effects. Lower down, it will affect things like the GI tract and the legs. Higher up, it'll be things like respiration, using your mouth and throat, arms, fingers and hands. All of these things are based on communication between the brain, the spinal cord, and the rest of the body. If you can find a way to mitigate some of the effects of damage to the spinal cord, you can bring back a lot of quality of life for people at a minimum. This is where a new implantable device comes in. This is intended to help with chronic back pain. Specifically chronic back pain that won't respond to medication, but chronic back pain. Something that's often a byproduct of deterioration of the spine in some cases, and in others, direct physical damage. The way it works is to continually send out small pulses of electricity. These stimulate the spinal cord, and they help to somewhat mute the sensation of pain. Previously, accessing the spinal cord in a way where you could do this would be incredibly invasive and dangerous, and most of the time, the rewards were not worth the risk. This new design device is far easier to implant, much less dangerous, and provides the same sort of reward we'd seen from past devices. That means you get all the benefit and very little of the risk. In the past, in order to get the necessary relief into the spine, you would have to remove part of the spinal vertebrae. This is not ideal. After all, putting holes or taking out large sections of bone is going to be painful regardless. but on top of that, it's very invasive and runs the risk of damaging the nerves within it. This new device can be made to an incredibly small size. In fact, it can be rolled up to 2 millimeters and thereby fit inside a needle. This means it can be inserted between the vertebrae and into the spinal cord, where it begins to provide the relief we've mentioned. Going now from clinical research to a case study, let's examine why you might need pain relief. This case study involved a 29-year-old Taiwanese woman who, strangely unknowingly, had chopstick fragments embedded in her sinuses for a week. This followed a fight with her sister. Nothing quite like sibling love. This fight was to have more significant repercussions than was immediately obvious. The first and most obvious of these is that 2 chopstick fragments wound up embedded in the nasal cavity. This was not immediately obvious, as nothing was showing up on X-rays. Looking into it further, it became apparent that yes, there was something solid and foreign in there. It's somewhat fortunate that how the chopstick fragments wound up where they were is actually the same approach taken in certain surgeries. That means they're able to remove them with relatively little worry. The 2 fragments aren't small either. Calling them fragments does seem a misnomer. At 3.5 centimeters, or roughly 1.5 inches, and 5 centimeters, or 2 inches, the 2 fragments aren't exactly fragments, more like large parts of the chopstick. Fortunately, removing them was relatively easy, and the person appears to have had no further complications, although we can't speak to their family relations after this particular adventure. Going back to clinical research, but in this case also animal research, the creation of a mouse embryo with a beating heart. That alone isn't something novel. Embryos with a beating heart aren't a new thing in research and are often used in developmental biology to understand what's going on. What's unique here is that it was grown in a petri dish, completely outside the organism in which it was going to develop normally. That's something interesting and unique at the moment. The reason this is unique and special is that the uterus or other location in which the embryo is going to develop needs to be incredibly complex. That's because you need to have the right signals at the right time, in the right concentration, and in the right place. Everything has to line up exactly right. If it doesn't, things don't happen in the right way. This is how you get incorrect development. Limbs don't grow correctly, cells don't differentiate. There are all manner of problems that can occur. In order to get the mouse embryo to grow, they had to do all of this, and not necessarily manually, but in a way that would allow the embryo to continue developing. Of course, being in the early days of research into doing this outside of another organism, the process is by no means perfect. In fact, there are some rather significant missing parts from this mouse, notably part of its brain. Developing a model like this of an organism's development in embryo is important to understand what's happening in pathologies, notably those that are present in both animals and humans, where the embryo is not developing correctly. Being able to observe what's happening and either recreate it, or in cases where you're looking at a disease, recreate the disease that is happening, you can then identify exactly how and what needs to be done in order to solve the problem later on when it occurs in a clinical setting. In other animal-based research, there's interesting exploration as to why camels can go without drinking for weeks. It's generally understood that camels store water in the hump in their back. Although not strictly true, it's a fair approximation of what's happening. At a more complex level, the camels have a whole raft of modifications to their body that allow them to survive without water. Everything from the way their body is shaped, how they breathe, what their blood is like, and so on. All of these help to drastically reduce the amount of water the body loses to the environment and how well it holds on to what it has. Among the hundreds of genetic changes that are turned on and off during dehydration, there are a few notable examples that come out in research recently. One of these regards cholesterol, and the role it plays in dehydrated camels. The exact way this works isn't clear yet, but the researchers postulate that by decreasing the amount of cholesterol in the membrane of cells, it would allow the better movement of solutes and water across the membrane of the cells. This allows water to be reabsorbed much more efficiently, and therefore they lose less into their kidneys and out into the urine. Going now across to plants from animals, we'll look at a weird and, frankly, somewhat disturbing fungus. This is a fungus known as the tea tree finger. The name comes from its rather obvious resemblance to, well, fingers. The fingers are part of the fruiting body. It shows up above the ground from the rhizome underneath. This rather bizarre and exotic-looking fungus is endangered. This status comes from a number of factors. Perhaps one of the most immediate and obvious is that it can be found on the mainland of Australia, not the most hospitable of places in the world. In fact, especially not hospitable for this fungus. It likes to grow around dead wood that's not been burnt for 30 years or more. Large parts of Australia undergo annual fire, and so the fungus cannot grow in those places. It must also have another symbiotic fungus that helps to break down the fallen timber. In another and somewhat environmentally related piece of news, we want to talk about Canada's wildfires. Notably, some of their bizarre behaviours that we're seeing as a consequence. The Canadian fires at the moment are not particularly great. I mean, relative to other countries, they could be a lot worse, but still, the last thing you want in your own backyard. What's interesting is that the heatwave, along with everything else, is creating clouds that are otherwise known as fire clouds. These clouds are unique from others in that they are generated by both the heat and moisture coming from the smoky wildfire. This is different from the normal process where light heats water, causes convection, and generates clouds that way. The fire clouds are important in that not only do they generate thunderstorms and lightning, they also create downbursts. This means the water is separate from the lightning, and therefore the lightning can create fire. The downburst also spreads embers from the fire across large areas. This creates two ways that the fire clouds can generate a new fire from the first. It is an interesting and unexpected byproduct of the naturally occurring phenomena, and one that's worth understanding and being aware of. Going now to our final news item for this week. Interesting discoveries on Mars. The discovery of methane. And no, the Curiosity rover is not farting, nor are there any cows to be found on Mars, and this contributes to the mystery. Methane is perhaps the simplest organic molecule. It is a single carbon with 4 hydrogens around it. It makes up a fair amount of our atmosphere, and in the long term, it could be an important mechanism for finding life elsewhere. It's possible that the methane being generated is coming from a geological process or some sort of microbial life form that's undergoing metabolism and using the limited available oxygen and other gases on the planet and something like the soil. The problem has been figuring out just when the methane occurs and where. Other researchers haven't been able to find it in the atmosphere, and that meant that it was possible that the detection was a misidentification or something similar. After going through all of the times it was detected and figuring out what was occurring when and how and why, they figured out it appears to be something to do with the nighttime and the lack of sunlight. Unfortunately, all this has really done is further create mysteries as to where it's coming from and why it's not disappearing or gathering at the rates that are expected. that's all the news we have for you this week. Thank you for watching. If you found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions that you have below